My project was the investigations of nucleotide modifications in Winthrop's bacteriophage collection, and I did this with the help of my mentor, Dr. Victoria Frost. Bacteriophages, also known as phages, are viruses which infect bacteria. They consist of a capsid head, which holds their genetic material, and a tail, which aids in the transfer of their genetic material into their bacterial host. Bacteria and phages have been involved in a billion-year co-evolutionary relationship where each have evolved to create their own defense mechanisms for protection. Understanding this co-evolutionary relationship is important since phages have increasingly been used in a number of different fields. One such field is therapeutic uses to help with the treatment of antibiotic-resistant bacteria known as superbugs. Bacteria have multiple defense mechanisms, and one such mechanism is the restriction modification system. This system utilizes restriction endonucleases, also known as restriction enzymes, to come cut up the phage's genomic material at specific sites known as the recognition sites. At the same time, bacteria must protect their own genome and utilize methyltransferases. Methyltransferases come to the bacterial genome at these recognition sites and methylate these sites so the restriction enzymes cannot cut up the bacterial host genome. At the same time, phages have evolved to create their own defense systems through modifying their DNA. These DNA modifications can be methylated sites, but more commonly, you will see that phages will modify nucleic acid bases. These modifications arise through biosynthetic pathways where side groups are added to the cyclic compounds during DNA replication. And these addition sites are shown in green on these cyclic compounds. Now that we understand how phages protect their genomes from restriction endonucleases, this leads to the objective of this project, which is to identify possible modification sites on bacteriophage genomes using restriction enzyme digestion. To achieve this, we first went to PhageDB to look at the sequence phages from Winthrop's phage collection. This database is full of phages found by students a part of the Science Education Alliance, sponsored by the Howard Hughes Medical Institute. Here off this database, we got the previously done digest and downloaded the genomic sequence of Winthrop's phages. We then utilized another online tool on New England Biolabs where we generated virtual digests where we could create comparisons between the previously done digest and the virtual. If this comparison suggested that phages may have had modification sites in their genomes, we then went back to the bench and re-extracted DNA from these phages. Then we recharacterized these phages' genomes using the same enzymes used on the previously done digest, but we also included two new ones. The first one was DPN1, which is a methylation-sensitive enzyme that cuts at methylated adenine sites, and the second one was MSC1, which is a methylation-insensitive enzyme, which cuts at any methylated site on the genome. After this digestion was complete, we then ran it out on agarose gel, and then we analyzed. Our first observation was that few phages were not resistant to restriction endonuclease activity. This is phage calcol, a part of the EG cluster. On the actual digest, you have your 1 kb ladder to serve as reference for us, and the uncut lane that served as a control. And then across the digest, you have your restriction enzymes. Looking at the virtual digest, you see that BAM-H1, CLAW1, ECO-R1, and HEN3 all did not cut this genome due to the fact that this genome did not have the recognition sites for these enzymes, and this is reflected on the actual digest. You can also note that HEY3, NSP1, SAC2, and SAL1 all had patterns for their enzymes, and these patterns are also reflected on the actual digest. Our second observation was that phages within the EA cluster demonstrated resistance to endonuclease activity. This is phage Liathus, a part of the EA cluster. Once again, we have our 1 kb ladder in our uncut lane and our enzymes across the gel. When looking at the virtual digest, you see that each enzyme should have created a pattern. But when we look at the actual digest, you see all enzymes, except for MSC1, reflected the uncut lane. This suggests to us that modifications are occurring at these recognition sites for these restriction enzymes. I would also like to note that MSC1 is a methylation insensitive enzyme. So seeing that there is a pattern here, this suggests to us that Liathus has at least one methylated site within its genome. This is phage IXL, also part of the EA cluster. When looking at the actual digest, you see that this digest is full of blocked restriction sites. This means that IXL also has modification at these recognition sites for these enzymes, and HEY3 is the only one that did not have a modification, since this is the only one that produced the pattern. Seeing that these phages within the EA cluster have similar blockages to their enzymes, this should suggest to us that phages within the same clusters may have similar or the exact same modifications, at least for the enzymes that we used. To further support this, this is a phylogeny tree from Winthrop's phages collected over the past 6 years. This helps prove our idea that phages within the same clusters are closely related, thus meaning that they may have similar modifications within their genome. This leads to our future directions. We hope to incorporate a sequencing protocol into our methodology to help identify modified bases. We also hope to compare genomes of phages within the same and different clusters to identify if DNA modifications are consistently observed. Bacteria and phages outnumber most biotic entities on Earth. Understanding how phages interact with their bacterial host are important since very little is known about this interaction. Though this research is in its preliminary stages, we hope to contribute knowledge to this vast but highly secretive relationship between phage and host. Thank you. I'd like to acknowledge my mentor, Dr. Victoria Frost, and the research team which I worked with this summer, our C collaborator, Dr. Maria Gainey, the Winthrop McNair program, and my director, Dr. Cheryl Fortner, for giving me this opportunity to participate in this wonderful program, my 2022 cohort that supported me throughout the summer, and of course to the Winthrop University Biology Department, HHMI, and SC Embry for any resources they contributed to the completion of this project. These are my selected references, and once again, thank you.